Hello everyone, welcome back to the session. So in the last class we have discussed about this long answer questions in the human excretory system as well as the human reproductive system. Okay, hope you have listened that. So now in today's class we are going to discuss about the long answer questions related to this genetics chapter, and it is very important. Okay, so we are going to start. The first question will be, so what are multiple alleles? Describe the multiple alleles with the help of this ABO blood groups in man. Okay, so we have to separate this question into two parts. The first part will be, we have to explain what is multiple alleles. Okay, and the second part of the question will be, we have to explain this multiple allele with the help of this ABO blood group system, because ABO blood group system is a best example for this multiple alleles. Okay, so first you should know what is alleles. Okay, so alleles is a different form of a gene. Okay, so see this particular H is responsible for this height. Okay, so see, so this is a chromosome. In this chromosome, this particular segment is responsible for the height. Okay, so this is a gene responsible for a height. Okay, so this height, it may be tall or short. Okay, it may be tall or dwarf. So this different form of a gene is called as an allele. Okay, so you should know allele always allele always expressed in pairs. Okay, so generally a gene has two alternative forms at the same locus on a homologous chromosomes. So homologous chromosome in the sense structurally and functionally it will be similar. Okay, now so it will be in same size and in the same function, so it has to be present at the same position. Locus means same position. So at this homologous chromosome, in the same position, they are having two different forms. So this is a gene, they are exhibiting two different forms. Okay, so sometimes the gene has more than two alleles in a population. Okay, see, so as I have mentioned you before, there is a two different form of a gene, that is tall or dwarf. Okay, but sometimes it may be more than two alleles. Okay, so that is called as a multiple alleles. Okay, see, a gene which is having more than two alleles in the population, that is called as a multiple alleles. Okay, a diploid individual carries only two alleles at a time, but multiple alleles are observed at the population level. See, you should understand the difference between these two. Okay, so a diploid individual, an individual can carries only two alleles at a time, but in a population level we can see the multiple alleles. How? Okay, see, just listen to me. The number of possible genotypes from a multiple alleles, we have to calculate this multiple alleles in the formula n of n plus 1 divided by 2. Okay, n into n plus 1 divided by 2. So this n is nothing but a number of alleles in a population. Okay, so this is what a multiple alleles. Multiple alleles in a sense, a gene has more than two alleles in a population, that is called as a multiple alleles. It is not in an individual, it is in an overall population. Okay. Now, so we have to explain this multiple alleles with the help of this ABO blood group system. Okay, so generally multiple alleles, the classical example of this multiple alleles in humans is called is the ABO blood grouping system. Okay, why is that so? Okay, first it has been proposed by this Carl Landsteiner. Okay, he won a Nobel Prize in 1930. Okay, what did he observe? Okay, both based on the presence or absence of the antigen. So we have two different types of antigens, A antigen and the B antigen. So whether which antigen is present, based on the antigen present or absent, we are determining the bloods. Okay, so we are determining the type of blood group. Okay, but before that, where it is present? In the membrane of this red blood cells. Okay, RBC. Okay, so consider this as an RBC cell. So in the RBC cell there is a membrane. So on this membrane with antigen, if it is an A antigen, we are saying this particular blood group as an A blood group. If it is a B antigen, we are saying this as a B blood group. If both the antigens are present, we are saying that as an AB blood group. If no antigen are present in the RBC, we are saying that particular blood group as an O blood group. Okay, so based on the presence or absence of the blood group, we are determining the types of blood group. Okay, Okay, so these antigens are carbohydrate groups. So majorly these biomolecules can be categorized into three, that is carbohydrates, proteins and lipids. Okay, so but this blood group system, it will be coming under which type? These antigens are coming under the carbohydrate groups. Okay, again that will be attached to the RBC membrane. See, membrane, we know that this membrane is made up of the lipid bilayer. So we have studied, I think, hope you all know that the plasma membrane is made of a lipid bilayer fluid mosaic model. Okay, so in that model they have the Singer and Nicholson have explained that this plasma membrane is made of lipid bilayers. Okay, so on this lipid bilayers this carbohydrate group has been attached, right? Now what are all the alleles are getting involved in this particular gene? Okay, the gene I. Okay, so what they have named the gene I. Okay, so they are located on which chromosome? Chromosome 9. So all these are important keywords. Okay, they are generally having three allelic forms. What is the allelic forms? For the antigen A, the genotype will be IA. The gene will be IA. Okay, the IB is responsible for coding the antigen B. Okay, so it is small I or IO. Okay, so small I or IO, both are same. Okay, so no antigens are produced for this particular gene. Okay, so that is why we are saying this I or IO will be recessive form. This IA and IB will be dominant form. Okay, so dominant relation. So that is why IA is equal to IB, because both are dominant. Okay, so greater than I, and this is recessive in condition. So this is how the equation. Okay, so IA is equal to IB greater than I. So this is what an equation. Okay, so as both IA and IB are dominant, okay, if both are dominant, that particular person having the AB blood group. Okay, so see, just listen to me. If IA and IB are present, that particular person, they are having the AB blood group. Okay, so as both IA is also expressed, IB is also expressed together, they are having the AB blood group. As both are expressed together, we are saying that particular term as a codominant. Okay, so this is what a codominant. So both are expressing without disturbing each other. Okay, so both are dominant over the I. Okay, so the best example for multiple alleles is also the blood group system, and for codominance is also the best example will be the multiple ABO blood group system. Okay, so this is what the genotype and the phenotype. Okay, so just listen to me. So if a person having IA and small a, okay, or IA, okay, so if this is a condition, that particular person is having this A blood group. Okay, so IA. So as I have mentioned you before, alleles always expressed in pairs. Okay, so IA. Okay, so that phenotype will be, the blood group will be the A blood group. Okay, and what will be the, so if it is an A blood group, what will be the name of the antigen present in the RBC? That will be the A antigen. If A antigen will be present, then that blood group type will be the A blood group. Then what will be the antibodies in the plasma? So the antibodies secreted by the in the plasma will be the antibody B. Okay, so in the blood group A, that person is having the A antigen and the B antibody. Okay, now in the B type of the blood group, so the genotype will be IB and IBI. Okay, two genotype is possible for this particular term. Okay, two genotype is possible. Now, what will be the antigen present in the RBC? So, if it is a B type of blood group, definitely there will be the B antigen is present. B antigen is present, and if it is which antibody, that will be the antibody A. Okay, the antibody A, right? Now, so if it is an AB type of blood group, that will be IA and IB. Okay, that IA and IB. So definitely this is a genotype goes for with which antigen? AB. Okay, so A antigen and the B antigen. Then what will be the blood group? AB type of blood group. As both antigens are present, there will be no antibodies present in the plasma. This is an important thing. And there is a small a, okay. It may be write like this or IO. Okay, both are similar. Okay, both are the genotype. It is not both, either small I or IO or IO. So both are the genotype for this O blood group. Okay, and the antigen present on this RBC in case of O blood group, there will be no antigen in this blood type, and but there will be the antibody. A antibody and B antibody, both the type of antibody is present in the plasma. Okay, so 1, 2, 3, 4, 5, 6. Okay, so for we are having generally 4 blood groups, which means 4 phenotypes. For these 4 phenotypes, we are having 6 genotypes. Okay, so this is an important thing. Okay, now what will be the significance? This inheritance shows multiple allelism and codominance, as I have mentioned you before. So the best example for the multiple alleles is the AB blood group system. And see this codominance, so this IA and IB. Okay, so both the blood groups are expressed together, so that is called as an AB blood group. So this is codominant in nature. Okay, without disturbing each other, both the blood groups are expressed together. Now, important in blood transfusion compatibility. See, if a particular person is having an A blood group, if he is met with an accident, definitely he has to he has to include with the blood group type person who is having the A blood group or the O blood group. Why is that so? Okay, so consider the person is having a blood group, okay, and this person is having a B blood group. Okay, so now what will be the antigen present in this body? B antigen will be present, and here it will be A antigen will be present. Okay, so now he is a donor. Okay, he is a donor and he is a recipient. Okay, so he is having an A blood group and he is having a B blood group. Okay, now see this A blood group, a person having the A blood group, he is having an A antigen, and B blood group having the B antigen. Okay, now whenever a donor, okay, he is a donor and the A blood group, he is giving his blood to the person having the B blood group. Now the antigen present in this A blood group will be entered into the body of the person having the B blood group. Now what is the name of the antibody present in the B blood group in the plasma? That will be A antibody. Okay, so as the A antibody is present in the person having the B blood group, as soon as the A antigen enter into the body of this person having the B blood group, this A antibody will fight against this antigen and it causes the agglutination. Okay, so this is an important thing you have to keep in mind, right? Now see, so this is a scenario. Okay, so just listen to me. So the person having the A blood group having the A antigen. Okay, he is having an A antigen. If a person having the B blood group, he is having the B antigen, isn't it? Now, so if this person donating his blood to the person having this B blood group, so as soon as it is entering, its A antigen enter into this person, the A antibody will be present in this person, will fight against this A antigen. So as soon as immediately it leads to the hemolysis or it leads to the agglutination. So what if in case of this AB blood group? So consider this person is having the AB blood group. Okay, and this person is having one person is having an A blood group, one person is having a B blood group, one person is having an O blood group, one person is having an AB blood group. Okay, so if AB enters into this body, there will be no problem. Okay, so as this is a person having an AB blood group and he is a recipient. Okay, so now what will be the antigen present in the this body? Okay, both A antigen is also present and B antigen is also present. Okay, so now there will be no antibody present here. There will be no antibody present in case of person having the A blood group. Okay, and there will be no antibody, no one is there to fight against this any antigen. Okay, so A blood group can also easily enter here, B blood group can also enter, O blood group can also enter, and AB blood group can also easily enter, as there will be no antibody secreted in the body of the person having the AB blood group. Okay, now what if in case of this O blood group? Okay, so as they can receive the blood from all the persons having all the type of blood groups, this AB blood group is considered as an universal recipient, right? Now, so O blood group, so this is a person having the O blood group, and what will be the antigen present here? There will be no antigen present here, but there will be both the antibody. A antibody will also be present, B antibody will also be present in case of person having the O blood group. Okay, so if A blood, so this is a person having the A blood group and B blood group, AB blood group, AB blood group is also there, and O blood group is also there. Okay, four types of person is there. Okay, so if there is an A antibody present here, okay, so if this is an A antigen will be present in this, and if the A antigen enter into this body of the person having O blood group, immediately after it is entering into this body, the A antibody, so which means the A antibody secreted by this B antigen will be leads to the agglutination. Again, if it is enter as the B antibody is also B antibody is secreted by this B antibody will be immediately fight against this blood group. Okay, if both the antibody, if A and B is not go separately, if AB will also if AB person will give the blood to this O blood group type, immediately after that leads to agglutination faster. So only this O blood group can give the blood to a person having the O blood group. So but he can give the blood, the O blood group can give the blood to all the person as it is having the no antigen. Okay, as it is having the no antigen, no antibody can fight against this particular antigen because no antigen is present. So he can give the blood to A blood group, person having an A blood group, B blood group, AB blood group, as well as the O. Okay, so as he can give the blood to all the types of blood groups, so he is considered as an universal donor, right? So this is what a blood group, this is what a multiple alleles, and this is what a ABO blood group system. Now comes the second question. So the second question will be the chromosomal theory of sex determination. Okay, the chromosomal theory of the sex determination. So what is sex determination? Okay, who is the determined decider of this sex? Okay, so first of all you should know the basic understanding of the chromosomes. Okay, so we are having the 46 types of 46 number of chromosomes, isn't it? Now also you should know that that is diploid in nature. Okay, so if it is a pairs, 23 pairs of chromosomes will be there in the human body. Okay, so in this 46 chromosome you should know that 44 chromosomes are considered as an autochromosomes. Okay, and 2 chromosome is considered as a sex chromosome, or we can call them as an allosomes. Okay, so this is a basic understanding. And if it is in a pairs, 23 pairs of chromosome is considered as an autosomes. Okay, and 1 pairs of chromosome is considered as a sex chromosomes. Okay, so this is a basic understanding. Okay, so in case of human beings I'm saying, okay, 46 total number of chromosomes, and if it is in a pair it will exhibit in the 23 pairs of chromosomes. Okay, so now what is sex chromosome? So as it is a chromosomal theory of sex determination, we are taking that particular sex chromosomes. Okay, a pair of chromosomes responsible for determining the sex. Okay, a pair of chromosomes, which means that 2 chromosomes or 1 pairs of chromosomes. Okay, so the chromosomes other than the sex chromosome is considered as an autosome. So we know that. And what will be the sex chromosome? That is nothing but an X chromosome and the Y chromosome. Okay, X chromosome was first identified as an X body by the scientist called the Henking. Okay, so Y chromosome can be identified by the Stevens and the Wilson. Okay, and this is sex determining, which means what? If Y chromosome is present, that particular body is considered as a male. If it is absent, it is considered as a female. Okay, so this is what a decider of the sex. Okay, so basically we can classify this sex determination as a heterogametic. Okay, why? Heterogametic. Okay, so the heterogametic sex produces dissimilar gametes. Okay, see, in case of humans it will be XX in case of female and XY in case of male. Okay, if XX is present, there will be no difference. Okay, it will leads to the female. Okay, so if it is an XY, so this is the one heterogametic. Homogametic means both the chromosomes are same, both the alleles are same. Here both the gametes are dissimilar, it is not same. Okay, so as Y chromosome is present here, as it is a heterogametic, so this is considered as a male. So this is what a heterogametic sex determination. Okay, sex of offspring is determined at the fertilization depending on which gamete fertilizing the egg. Okay, so consider this as an organisms. Okay, so XY and XX. Okay, so if X is going to fuse with another X, it gives the XX, that is female. Okay, so if X is going to fuse with another X, it gives the heterogametic male. Okay, so it is based on which gamete is fertilizing the egg, the sex is getting determined. Okay, so this is what heterogametic sex determination. Okay, now let me explain you in detail. Okay, just listen to me. See, sex determination. Okay, so this sex determination we can classify into two. Okay, male heterogamety and female heterogamety. Okay, male heterogamety, female heterogamety. We can further classify this male heterogamety into two. Okay, so what is that? XX, XY, XX, XO. Okay, so and female gamety, heterogamety, we can further classify them into two. Okay, so ZZ, ZW. Okay, so ZZ, ZO. Okay, so this is what two types of sex determination. One is male heterogamety and female heterogamety, which means I'll explain you. Okay, so here now the best example for this will be the humans. Okay, human as well as the Drosophila, that is the fruit fly, Drosophila and the humans. Okay, the example for this XX and XO will be the grasshopper. Okay, and ZW will be the birds. And ZZO will be the domestic chickens. Okay, there are also some other examples, kindly go through it. Okay, I'll take this one examples for your better understanding. Okay, now see here, so this is in the male heterogamety. Male heterogamety means what? Males are producing different gametes, so we can easily identify whether this XX is a male or XY is a male. Definitely XY is a male. Okay, because we are saying this particular term as a male heterogamety, and here there will be an absence of one X. Okay, so that is what another male heterogamety. So this XO will be the male and this XX is the female. Okay, now another important thing will be this EXX EXY. Okay, so if X is fused with another X, it results in the female. Okay, so if the X is fused with the Y, it produces the male. Okay, so if X is fused with another X, it gives rise to an X, and if it is fused with the Y, it results in the male. Okay, so based on the presence or absence of this if this Y, we can say this particular is male or a female. So you should know in the male heterogamety, the decider of sex will be the male. Okay, why? Because Y chromosome is present here. Okay. So like in the same way, this XX and EXO. So if the X is fused with an X, it gives the female. If the X is fused with the O, it gives the male. In case of a grasshopper, right? Now in the birds, just come here. Female heterogamety. Female heterogamety in the sense, this Z is a female or a male. This ZW is a female or male. So female heterogamety, which means they are producing the different gametes. So obviously this ZW is considered as a female. Okay. So now based on the presence or absence of this W, we are going to say whether this is a female bird or it is a male bird. So if the Z is fused with the Z, they are gives rise to the male bird. Okay, if the Z is fused with the W, they are giving the ZW, that is the male. Okay, so that is a female. Okay, so here in female heterogamety, the decider of sex will be the female. Okay, so in birds, the question can be asked in very short answer question. So here the decider of sex in case of female in birds will be the female. Female is the decider of sex in the birds. Okay. So now like in the same way in the domestic chickens, if the Z is fused with the other Z, it gives rise to a male. If the Z is fused with the O, there will be no chromosome, no gametes that present here. It results to male. So that's all about the sex determination. I'll explain you in little bit detail with the help of this theory. Okay, see, male heterogamety. Males are producing dissimilar gametes, so that is why XX or it will be EXO. That is what a dissimilar gamete. So we are saying this particular as the male heterogamety. Okay, females are producing the similar gametes. Okay, it may be XX. Okay, so the first type will be the XXXO type. It is present in some insects, that is grasshopper, cockroach, bugs. Okay, so these are all the examples for this particular male heterogamety. Now the female XX, that we are saying this, they are producing the same gamete, that is homogametic. Male, that will be the XO, the different gamete, so that is why heterogamety. Now, in the female gametes, all are X. In the male gamete, it may be X or O. Okay. So, the offspring sex depends on the sperm type, because whether it will be the X or the O, which is going to fuse with the XX, they are deciding the offspring sex. Okay. So, here it will be the male. Decider of sex will be the male. And the second type will be the XX and XY type. Okay, it is found in humans, Drosophila and most of the mammals. We know that XX will be the homogametic and XY will be the heterogametic. Okay. Now, here the female gametes is only one, that is X, and the male gametes will be the X or Y. Okay. So, the fertilization, that will be the X sperm. Okay, fertilization by X sperm to the female, by the Y sperm to the male. Okay, so this is the very important thing. Whatever things I have explained you there, the same thing will be reflected here also. So see here, so this is a female. Okay, so that is this AA is nothing but an autosomes. Okay, so here the sex chromosome will be the XX and here the sex chromosome will be the XO. Okay, so we can say that as an AX and here it will be the AX and EA. Okay, so this EA, so if this particular thing is fused with this EAX, we can say this as an AAXX. And if this X fused with this, we can say that as an AAEXO. Okay, so based on this thing, we can determine the sex. Okay, now Homo sapiens AAXX and AAXY. Okay, as it is producing the, so both are A, we are taking one A, both are X, we are taking one X. Okay, but here it will be the different gametes, so we can say that as an AX and EAY. Okay, so two gametes are present. Okay, so if that is an AX and AX, again it will be gives rise to AAXX. This is an AX and this is an AY, so it gives rise to AA and XY, and this will be the male. Okay. Now, The female gamete, female heterogamety. As the females are producing a dissimilar gametes, we are saying that as in female heterogamety, male are producing the similar gametes. Okay, so the first type will be the ZO and Z type. Z type. So what in which are all the insects we can see this type of heterogamety? So in moths and in some of the butterflies. Also the domestic chicken. And ZO, that is considered as a heterogametic, ZS is considered as a homogametic. Why? Because both are similar gametes. Here it will be different. Okay, so female gametes are Z or O, but male gametes are only Z. Okay, so here the decider of sex will be the female. Okay, now the second type in the female heterogamety will be the ZW and ZZ. Okay, so where this can be present, it is present in birds, reptiles and fishes. Okay, so here ZW will be the heterogametic, so that is why we are saying this as a female heterogametic. And males here it will be the ZZ homogametic. Okay, so the female gamete, that is Z or W, male gamete, that is Z. Who is the decider of sex here? Again it will be the female. Okay, so see here, AAZO. Okay, and here it will be AAZZ. So this will be the male and this will be the female. So as here that will be, it is fused with AZ and there will be no gametes, it is just A. Okay, and here it will be both are A, both are Z, so we can take that as an AZ. Okay, so if this AZ is fused with this AZ, it gives rise to AAZZ. Okay, so it obviously it will be a male. Okay, so if this A is fused with this AZ, so we can say that as an AAZO, and this will be the female. Okay, now in the birds, so just see here AAZW, this is a female, and here it will be AAZZ will be the male. Okay, as it is both different, this AA we are taking one AZ and AW. Okay, so this is what a gametes. Now here both are A, both are Z, so we are taking one AZ. Okay, so if this AZ is fused with this AZ, we can come up with the AAZZ. So if AW is fused with the AZ, we can say AAEZW, and this will be the female. Okay, this is in case of birds, right? Now, so that's all about the sex determination in the female, the sex determination of our animals. Now, another important thing will be the criss-cross inheritance. What is criss-cross inheritance? Explain the inheritance of one sex-linked recessive character in the human beings. Okay, so we have to explain what is criss-cross inheritance. What is criss-cross inheritance and what will be the inheritance of one sex-linked, how it is getting inherited with another. Just it, okay? Now, criss-cross pattern of the inheritance. Okay, so this criss-cross pattern also skip generation inheritance, also called the skip generation inheritance. It is skipping the inheritance. That is what a criss-cross pattern. Okay, so it occurs when a sex-linked recessive gene is transmitted from the male parent to his male grandchild through a carrier female of the first generation. So here you have to look at many important things. The first things will be this criss-cross pattern. Why we are calling this particular as a skip generation inheritance? There is a reason. Okay, as because it is a sex-linked recessive gene. Okay, first thing will be the it occurs only when there is a sex linked in a recessive gene. See, as I have explained you before, we are having 44 chromosome, we are having 45 and 46, two chromosome, that is sex chromosome. Okay, so if there is any genetic disorder present in this particular two chromosome, we are telling them as the sex linked recessive sex linked disorder. That sex linked disorder, that may be dominant or recessive. Okay, see what I have mentioned you, we are having 44 chromosome, that is nothing but an autosome, and we are having two chromosome, they are nothing but a allosome, that is the sex chromosome. Okay, if any disorder occurs in this particular chromosome, we are telling that as an X-linked disorder, that X-linked disorder or sex-linked disorder. So that disorder may be either it will be dominant or recessive. Clear? Now, that crisscross pattern of inheritance that have been exhibited in the sex-linked recessive gene. Okay, and the second thing will be, so first point it will be the sex-linked recessive gene, and from the male parent, okay, this is a male and this is a female, so okay, both are getting married, okay, both are getting married and they are giving one child. Okay, so they are giving the one male child. Okay, so again he is getting married a female. Okay, again there is a one male child will be present. So from the male parent it has to be transmitted through the male grandchild. Here the female will be the carrier. Okay, always in a crisscross pattern, who is a carrier here? Female will be the carrier. Why? Because as it is in X-linked, we are denoting this particular sex as an XX as a female and XY as the male. Just listen to me very carefully. Okay, so as it is an X-related disease, if only one X is present in case of male, if this X has damaged or having this disorder, definitely that disease has to be transmitted to the next generation. But in the female, we are having two X. 2x will be present. If one is acting as a one is affected, another x is there to replace the function of this affected x. Okay, so here in case of male there will be no new x will be there to replace the function of this x. Okay, so this is an important thing. So that is why female is always acting as a carrier. Okay, so you have to understand from the transmitted from the male to the male grandchild. Okay, male grandchild will, female is acting as a carrier. Okay, so in the first generation, so all these words are very important. So the best example for this will be the color blindness. Okay, I'll explain you little more detail in the next slide. So just listen to me. So this is an normal female. Okay, so normal female. Okay, and here there will be the color blind male. Color blind male. If one X is getting affected, definitely he is transmitting this disease to the next generation. Okay, so XC and Y. Okay, this is how it will be represented. So now what will be the gamete? So X plus and X plus will be there. Okay, and here it will be the XC and the Y. Okay, so whenever we are crossing this, so there will be an X and EXC will be here, and here it will be normal X and normal Y. Okay, so here who is a carrier here? So this is a female and this is a male, and there will be no carrier, no affected gene is present in case of this X. Okay, so that is why we are having the normal male and the normal but carrier female in the first generation. Okay, normal but the carrier. So what if in case of the second generation? Okay, now just see here, so this X plus XC. Okay, so the X and XC, which is carrier female, but here it will be the normal male. Okay, so this normal male again it will be fuses with this X and this X. Okay, so that is a normal female, and if it is this X and this Y, it will be normal male. Okay, normal male. See here it will be XC and X, which means normal female but it is a carrier. Okay, normal but carrier female, and again it will be XC and Y. XC and Y, which means color blind male, because it will be XC and Y, which means there will be definitely he is getting affected because it is a male. Okay, he will not be acting as a carrier, he will be the affected male. So you should know that out of 4 child, 3 is getting affected, 3 is having the normal vision and 1 is getting affected. So we are having the ratio 3:1, 3 normal vision and 1 is getting affected. Okay, so this is what a crisscross pattern. Okay, so initially the male is getting affected. Okay, but the normal female is there. Okay, so in all these things, always the male is getting affected, the female is acting as a carrier. Okay, so here the colorblind male, in the first generation, the female is acting as a carrier, and in the second generation, the male is getting affected. Okay, male is getting affected. Now, the second thing. Here, the color blind female, if the female has to be color blind, both the X has to be affected. This is the important condition. Okay, and in the female, a normal female means, normal male means he will be EXY. Okay, now how the gamete will be EXC and XC. Okay, so this is how the gamete produced by this female. And what will be the gamete produced in this normal male? It will be X and Y. Okay, so if it is EXC and if it is CX, it will be normal but the carrier female, because XC is present, normal X is present. So normal but carrier female will be present. Okay, now here the XC and the Y. Okay, so here it will be XC and Y. So here definitely he will be the color blind male. Okay, so XC and Y, color blind male. Okay, now what will be the next generation? X plus normal X will be there, which means here it will be the carrier female. So one normal X will be there, so one color blind X will be there. Okay, so here it will be the color blind X will be there and Y will be there. Okay, so whenever we are using the CX, normal X and color blind X, so here it will be the normal but the carrier female, no doubt in that. And again a normal X and normal Y, we are getting a normal male. Okay, so color blind X, so color blind X with the color blind X, definitely it will be the color blind female will be there, and the carrier color blind X, and here it will be the normal Y, so it will be the color blind male. Okay, so all these are important, these are all the two important conditions. So in the first condition, so in this we have taken a color blind female with a normal male. So here we can take in the normal female and color blind male. Okay, okay, how the crisscross pattern works. Okay, so first just think this color blindness. Okay, so in the color blindness it is EX linked, and you have to know that will be the recessive disorder. Okay, so what will be the causes? Why the, what is meant by color blindness generally? Okay, so mutation, definitely the X chromosome is getting mutated. The X chromosome is getting mutated. Okay, so why in particularly in which segment of this X chromosome is getting affected? Okay, so which means in the color perception. Okay, so controlling the red and the green color. A person who is affected with this color blindness can't differentiate whether it will be the red color or green color properly. Whenever they are seeing the red color, for them it seems to be green color. Okay, whenever they are seeing the green color, they seems to be red color. Okay, so that is what a color blindness actually. Now, so what will be the inheritance? So whatever we have seen here in the theoretical points, just now we have, I have explained you in the flowchart point. Okay, see a normal female, that is X6, color blind male. Okay, XY. How do we represent this color blind man? Like this. Okay, X6, XY. All children are normal and the daughters are acting as a carriers whenever the woman is normal and the man will be the colorblind. Okay, so whenever there is a carrier woman, that is XC and X, that is a carrier, normal man, all the daughters are normal or the carriers. It may be normal or the carriers, and 50% of the sons may be colorblind. You can cross these particular two things. Okay, so the trait passes from the male parent, that is male parent, to the carrier daughter and to the male grandchild. Okay, this is what I have explained you in that previous thing. Now, and the second thing will be the hemophilia. Okay, so the first example will be the color blindness and the second example will be the hemophilia. So in the hemophilia, again it will be the X-linked and it will be the recessive disorder. Okay, so what is exactly the hemophilia? So hemophilia in the sense, so the differentiation, not a differentiation, if a particular person accidentally cut his hand, okay, within a few seconds or minutes, it has to be clotted, the blood has to be clotted, okay, but the person who is experiencing this hemophilia, if he is accidentally cut his hand, the blood will not stop. Okay, so which means what? There will be an absence of this clotting factor in the body of that particular person who is having this hemophilia, who is getting affected. And what will be the name of the clotting factor? That is nothing but a thromboplastin. And there is one more thing, there is a two types of the hemophilia, hemophilia A and hemophilia B. Okay, so if the hemophilia A, the lack of clotting factor 8, this will be more predominant, and the clotting factor 9. So generally there is a thromboplastin. So in the blood cells we are having three types of blood cells. One is RBC, and another one is WBC, and another one is platelets. This platelets is the one responsible for the blood clotting. Okay, so for this platelets to perform this clotting function, it has 13 important factors. Okay, from the factor 1 to factor 13. Okay, so factor 13. Okay, so from the factor 1 it has to convert to the factor 13, which is nothing but a fibrinogen. It has to convert it to fibrin. Okay, so this is acting as a clotting factor. And factor 4 is considered as a calcium. Calcium is also plays an important role in the blood coagulation. Okay, so you should know, if it is a hemophilia, the factor 8 is absent. If it is a hemophilia B, the factor 9 is absent. What will be the symptoms? Prolonged bleeding due to slow clotting formation. The clotting will be very slow. Okay, and the inheritance, again it follows the crisscross pattern, just like the color blindness. Now the third sex-linked recessive disorder will be the Duchenne muscular dystrophy. Again it will be the X-linked and recessive muscular disorder. Okay, so it will be 1 in 3,600 boys. Okay, so this is an important thing. What will be the causes? Mutation in the dystrophin gene, and the dystrophin gene will be present in the X chromosome. Okay, so if the dystrophin gene is getting affected or mutated, it is the problem where they are synthesizing the dystrophin protein. Okay, if there is a defective dystrophin gene, definitely there will be the defective in the dystrophin protein formation. Now what will be the symptom if the dystrophin gene is not function properly? Progressive muscle weakness and loss of coordination, rarely survive past early 20s. Okay, they can't survive. Okay, so what will be the inheritance? How they are inherited like this? If mother is a carrier, okay, 50% of sons is getting affected, all the daughters are expected to be normal. Okay, here the sons are getting affected, okay, than the daughters, because they are acting as a carrier here. Okay, so next thing, that's all about the X crisscross pattern. Now we are going to see about this common genetic disorders. Okay, and the first we are going to see about this Mendelian disorders, that is monogenic disorders. If there is a mutation in one single gene, that is called as a Mendelian disorder. Okay, so that is inherited in the Mendelian patterns, whether it will be the autosomal or it will be the allosomal. If it is an autosome, there will be whether it is dominant or recessive. If it is an allosomal, whether it is dominant or recessive. We have studied this dominant and recessive in the Mendelian genetics, where in the monohybrid cross, dihybrid cross, and trihybrid cross. Okay, so like in the same way they are inherited. As they are inherited, whether like in the monohybrid Mendelian genetics, we are calling this particular disorder as a Mendelian disorder. The example for this Mendelian disorders will be the hemophilia, sickle cell anemia, and the color blindness, thalassemia, cystic fibrosis, DMD, and albinism, phenylketonuria. We are going to explain once again, one by one. Okay, so in hemophilia it will be the X-linked recessive type. As I have explained you before, the factor, there will be an absence of this factor in case of hemophilia A. If it is a B, there is an absence of this factor 9. And C, hemophilia C, it will be the factor 11 deficiency. Males are getting affected more due to the presence of single X chromosome. In the female, there is 2 chromosomes. If your one X is getting affected, there is other X to replace the defective X. Okay, so female is always acting as a carrier and male is getting affected. It follows the crisscross pattern, carries mother to the sons, right? Now, and the second genetic disorders will be the sickle cell anemia. See, generally you should know that in the chromosome 11. Okay, so in the chromosome 11, this chromosome 11 is responsible for this hemoglobin. Okay, hemoglobin structure. Hemoglobin structure. So generally in the hemoglobin structure, it is a 4 polypeptide chains will be there. In the 4 polypeptide, 2 alpha and 2 beta will be there. So in the beta globin gene, there is a problem. So that is why we are experiencing the sickle cell anemia. Okay. So you should know that it is mainly because of the single gene, that is point mutation. So in the 6th position, okay, so this is an mRNA. Okay, so this is an mRNA. So in this strand, this is one codon and this is the second codon, this is the third codon, this is the fourth codon, this is the fifth codon and the sixth codon. So in the 6th position, okay, so GAG is a codon, okay, that has to be replaced by the GDG. Okay, so GAG is responsible for giving the amino acid called the glutamic acid. GTG is responsible for giving the amino acid valine. Okay, so you should know at the 6th position the glutamic acid is replaced by the valine, so that originally the shape of RBC will be biconcave, and due to the amino acid change the structure of this RBC will be converted into sickle shaped. So that is why the disorder called the sickle cell anemia. Alright, so just listen to me. At the 6th position, the codon GAG has been replaced by the GTG, so that the amino acid glutamic acid will be replaced by the valine, so that the shape of RBC has been changed. Okay, so that is why we are experiencing the sickle cell anemia. So RBCs are sickle shape. There will be low oxygen, not having the 4 polypeptide chains in the hemoglobin. So there will be the oxygen carrying capacity will be lower, and it is causing the pain and results in the organ damage. Okay, and the heterozygotes. How the heterozygotes condition, that is HbA and HbS. HbS means sickle cell hemoglobin. It is a carrier and it is partially resistant to malaria, which means what? Those person who are having the sickle cell anemia, they will not experience it with this malaria. Why? Because for this malaria, the Plasmodium parasite needs the RBC, proper RBC, okay, for the reproduction process. Okay, so if the RBC's shape is not proper, they can't survive here as it is in a sickle shape. There is no proper space for this particular parasite to complete the life cycle. So the mosquito malaria will not be experienced by the person who is having the sickle cell anemia. Right? Now, phenylketonuria. So it will be autosomal recessive. Okay, it is an autosomal recessive again, and here it will be the gene which is responsible for this will be this PAH gene. And what will be the name of the number of this chromosome? In the 12th chromosome. In the 12th chromosome, okay, so phenylalanine hydroxylase enzyme is getting hydroxylase gene. Phenylalanine hydroxylase gene. Okay, so this gene is responsible for producing the phenylalanine hydroxylase enzyme. Okay, so as this gene present in the chromosome 12 is getting affected, there is no point of giving the enzyme, that is phenylalanine hydroxylase enzyme. If this enzyme is not functioning properly or not produced in a proper amount, this phenylalanine is not converted into tyrosine. Okay, so if this chromosome 12, in this chromosome 12, if this PAH gene is not functioning properly, there is no production of this enzyme. PAH enzyme will be produced. If the PAH enzyme is not produced, the phenylalanine will not be converted into tyrosine. If the tyrosine will not be produced, the phenylalanine will be converted into phenylpyruvate. Okay, if the phenylalanine pyruvate it is accumulated largely, it leads to the mental retardation. Okay, so this is very important. Now, color blindness. So I have explained with this color blindness before. I don't want to explain this much. And thalassemia. Again, the thalassemia, it will be in the chromosome 11. It is in the chromosome 11, and there will be chromosome 11, and again it will be leads to the hemoglobin structure. Okay, it is in the hemoglobin structure. So it will be defect in the alpha globulin chains or the beta globulin chains. Again it will be autosomal recessive. So if it is in the alpha globulin chain is getting affected, that is considered as an alpha thalassemia. If the problem is in the beta globulin chain, it will be results in the beta thalassemia. Okay, so here hemoglobin, the HbA1, HbA2 are the genes present in the chromosome 16. So this is important thing. If it is in the chromosome 16, it is alpha thalassemia. If it is in the chromosome 11, it will be beta thalassemia, and we can call them as the Cooley's anemia. Okay, it results in the defective RBCs. Okay, the RBC will not produced properly. And cystic fibrosis. Cystic fibrosis, it will be autosomal recessive. Okay, again it will be autosomal recessive, and the mutation will be there in the chromosome 7. Okay, so the symptoms leads to thick muscles in the lungs, so that no diffusion will be occurred properly, no exchange of gases will be occurred properly, and the pancreas, gastrointestinal tract, it leads to respiratory and digestive problems. That is what a cystic fibrosis. Okay, so far we have completed this Mendelian disorders. Now what is this chromosomal disorder? Okay, so I'll explain you a little bit detail. See, so if there is a problem in the 44 chromosome, we are saying that as an autosomal disorder, and if the problem occurs in this 2 chromosome, that is chromosomal disorder. Okay, so first one will be this chromosome, sex chromosome. Okay, so allosomal disorder. Okay, so we are saying that as a Klinefelter syndrome and the Turner syndrome. Okay, so 44 chromosomes are very normal in case of this Klinefelter syndrome, and in the 45th and the 46th, that is the X and Y chromosome, or in the X chromosome, what is there the condition? Okay, here the X chromosome, it will be XXY. Okay, we can't say whether it will be the proper male or where there will be the proper female. Okay, so the male, they are having one extra X chromosome, so the total chromosome will be, so it has to be X and Y or XX. Okay, 44 chromosome and 45, 46. The 46 chromosome is balanced, and this male is having one extra chromosome, so it leads to 46, 45, 46, 47. So totally 47 chromosome, and the sex chromosome here will be XXY. So the male is having one extra X chromosome, so that particular male is having the female characteristic features: hypogonadism, okay, reduced fertility, gynecomastia, which means breast development in males, and rounded hips will be there in the Klinefelter syndrome. Now in the Turner syndrome, it is actually a female and one X chromosome is missing here. So 44 chromosome will be proper, and this is 45, and 46th chromosome will not function properly. So the total number of chromosome will be 45, and only one X chromosome will be present. That will be not complete female has been produced. Okay, so start stature, webbed neck, gonadal dysgenesis, broad chest. So these are all the symptoms of this particular Turner syndrome. Now, autosomal disorders in the chromosomal disorders: Down syndrome, Edwards syndrome, and Patau syndrome. Okay, so trisomy 21. Trisomy in the sense 2n plus 1. So in case of human beings, this 2n is nothing but a 46 chromosome. 46 chromosome plus 1. So totally 47 chromosome. Okay, so what is this 21? In the 21st chromosome, there will be one extra chromosome will be present. So that is why 47 chromosome, female plus 21. So symptoms will be short stature, flat face, and intellectual disability will be there. Now, Edwards syndrome. Again it will be trisomy 21. Trisomy 18. Here in the 18th chromosome, there will be one extra chromosome will be there. So totally 47 chromosome plus one extra chromosome will be there in the 18th chromosome. And what will be the defect? There will be a high fetal mortality. Okay, will be there. And Padar syndrome, again it will be 2n plus 1. In the 13th chromosome, an extra chromosome will be there. So as a result, 47 chromosome will be present in this particular individual, and it results in the heart or kidney defects, intellectual disability, and early death is also possible here. Okay, and another fourth will be the Credit-Chatz syndrome. Credit-Chatz syndrome in the sense, so in the chromosome 5, okay, in the chromosome, what is meant by P? Okay, so in the chromosome, so this is how the chromosome will be. This is a short arm, this is a long arm, and this short arm is considered as P, and this long arm is considered as Q. Okay, so as Credit-Chatz syndrome means, whenever this person is crying, it looks like a cat like cry. Okay, mental retardation and facial abnormalities will be there. Okay, so the deletion here, there will be deletion occurs, deletion in the chromosome 5. Okay, deletion of some genes in the chromosome 5, particularly in the short arm. Okay, and the chronic myelogenous leukemia, here translocation is there. Okay, translocation between the chromosome 9 and the 22. So this results in Philadelphia chromosome. Okay, so this reduces abnormal, produces abnormal tyrosine kinase, uncontrolled cell division. It results in the leukemia. Okay, so that's all about this common genetic disorders. We'll see in the next class. Thank you so much. Okay, so in this class we have completed this genetics long answer question. Okay.